Hello, welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the ninth lecture and we will discuss about super resolution microscopy. So what is a super resolution microscopy? So super resolution microscopy, it is also known as SRM, is an advanced optical microscopy that offers resolution beyond the diffraction limit. Okay. The resolution of conventional light microscopy is limited to around 200 nanometers due to the diffraction of light. So we learned about several microscopy techniques in previous classes about wide field microscopy, confocal microscopy, light sheet microscopy, and several other types of microscopy we learned, right? But in all these microscopy techniques, the resolution is limited to 200 nanometer due to the diffraction of light. And what does it mean practically? So here in this diagram, you can see what is the diffraction limited resolution of a microscope. So let's say you have an object of different sizes starting from 18 nanometer to 42 nanometer, 66 nanometer, 90 nanometer, and 138 nanometer. Okay. And if you see all this object under the microscope will produce the diffraction limited image of this object like this. Okay. So this is the diffraction limited image of the object of different sizes. And what you can see here, though the object size increases in size, okay, but the image size is not increasing because this is the diffraction limited resolution of a microscope. Anything below 200 nanometer will look like a 200 nanometer in diameter, okay, because the microscope produces the diffraction limited image of the object. Here you can see there are two objects of 50 nanometer lying close to each other. The microscope will produce the image like this because they are less than 200 nanometer and they are less than 200 nanometer apart. Okay, here there are four small spots of 10 nanometer each, but the microscope will produce the image like just one dot because they are less than 200 nanometer in size and they are 200 nanometer, less than 200 nanometer apart. Okay, so this is because of the diffraction limited resolution of a microscope. So super resolution microscopy that we will study today, they cross this barrier of diffraction limit, and we will study how those techniques crossed this barrier of a diffraction limit. Okay, so current super resolution microscopy approaches can achieve the resolution of around 10 times greater than the conventional light microscopy. So see, the conventional light microscopy, the resolution limit is 200 nanometer in X and Y direction. Okay, but the super resolution microscopy can resolve up to 20 nanometers. Okay, so in practice, that means that if the two fluorophores they are separated by 20 nanometers are visible as separate entities with certain super resolution microscopy methods, whereas they would need to be around 200 nanometer apart to appear as two separate spots in conventional light microscopy. Sometimes people consider confocal microscopy or some computer-aided deconvolution microscopy as a super resolution microscopy approaches because they can enhance the resolution up to 100 nanometers. Okay, but they are not the core super resolution imaging technologies. They just improve the resolution by either computationally improving your images or by the confocal mode of illumination. Okay, but they are not the real super resolution imaging. The real super resolution microscopy techniques, they are divided into three categories. The first one is known as structured illumination microscopy, known as SIM. The second one is the stimulated emission depletion microscopy, known as STED. And the third one is stochastic optical reconstruction microscopy, known as STOM, or the photo activated localization microscopy, known as PALM. Okay, so SIM, STED, STOM, and PALM. So these are the four words that we will discuss frequently today. Okay. So on this slide, you can see using the standard optical microscope, you can resolve the structures in this range. Like you can see the cells or cellular structures up to 1 micrometer using the standard optical microscope like wide field illumination or the confocal microscopy. But with the super resolution microscopy, you can resolve the structures up to 10 nanometers. Okay, and with the electron microscope, you can even resolve the structures up to 1 nanometer. But In the optical super resolution microscopy, we can go only up to 10 nanometers, not up to the 1 nanometer yet. Okay. So coming to the first type of super resolution microscopy, which is known as structured illumination microscopy, SIM. Okay. So SIM is used to increase the spatial resolution. Spatial resolution means the SIM technology can increase the resolution in X and Y direction, not in Z direction. Okay. And how it is achieved? So a fluorescent sample is excited multiple times using striped illumination pattern, 3 phases or 5 phases, and each time the orientation and position of the strips are changed. So let me explain you using a diagram. So see, this is the typical schematics of the structured illumination microscope, in which the illumination happens in this kind of a grid pattern. Okay, so what happens? Let's say this is your fluorescent sample, and the illumination is also in the grid. Okay, so this grid is rotated on top of your sample in this 3 orientation. So here the grid is like this. In the second sample, the grid is like this, the illumination grid. Okay, and in the third sample, the grid is like this. So basically, we are changing the angle of the illumination grid. Okay, and we are illuminating the sample using 3 different grids, and that produce this kind of a Moir pattern. Can you see this kind of a pattern here? The U-shaped pattern because of the gridding. So this is known as a Moir pattern, and then we use computation software to construct the super resolved image from this Moir pattern of the illumination. Okay, so let me explain you in this another diagram. This is a diagram to explain the same thing, but using different schematics. So let's say this is your cell sample, and these are some fluorescent molecules that you want to see in the super resolution mode. So now your this sample will not be illuminated altogether. Instead, it will be illuminated in this kind of a grid. Okay, and first you will put the these are 3 different angles of the grid, which are known as 3 phases. So you illuminate the sample with 3 different grids or 3 different angles, and then you use the computation software to reconstruct the image to produce the super resolved image of your structure. So here is the comparison of the image acquired by the wide field microscopy and the same image acquired under the structured illumination microscopy, and you can see this. You can see the better resolved structure using this approach. Okay. So coming to some of the limitations of this SIM microscopy. So typically it provides a resolution of up to 90 to 140 nanometer. See, if you remember, the diffraction limited resolution of any microscope is 200 nanometer, right? But using SIM, you can reduce that resolution to 90 to 140 nanometer in X and Y. There is no significant increase in resolution in the Y direction. The speed of this imaging is low because each sample or each position is imaged 3 to 5 times for this different gridding. Okay, so the speed of imaging is low. Okay, you need to take 3 to 5 images for the same sample, same position. The advantage is no need for spatial fluorophores. So let's say your sample was GFP labeled and you observed that sample under the regular wide field illumination microscope. The same sample you can see under the SIM microscope. The same GFP you can see under the SIM microscope. You do not need to label your sample differently for this structured illumination microscope. And the disadvantage of this method is that limited super resolution and excessive photo bleaching, because as you can see, there is a limited super resolution. It can produce give the resolution of up to 90 to 140 nanometer in XY, which can sometimes be achieved by the confocal microscopy or the deconvolution microscopy as well. Confocal and deconvolution microscopy can also achieve the XY resolution of up to 100 nanometers in certain conditions. Okay, so resolution wise, that is not significant enhancement, but still you can get better images. And another disadvantage of structured illumination microscopy is the excessive photo bleaching, because one image you have to take in 3 to 5 different orientation. So your sample will be illuminated 3 to 5 times to capture the image using 3 or 5 different grid patterns. So this is about structured illumination microscopy, and let us move on to the next type of super resolution microscopy, which is known as stimulated emission depletion microscopy, which is known as STED also, STED. So STED uses a technique called spatially patterned excitation. And during STED microscopy, two lasers are used on the focal plane. The first laser is known as the excitation laser, which is depending on your on the excitation wavelength of your fluorophore. You have to use different wavelength laser like 390 nanometer, 488 nanometer, 561 nanometer, or the 647 nanometer, depending on the fluorescent dye that you are using. And that is a kind of a regular laser that we use in other kind of confocal microscopes. Okay, and the other laser is known as the STED laser. Okay, so STED laser reduces the effective point spread function of the excitation laser. So how it works? So in the regular confocal microscope, you use this kind of a excitation laser only. So that this excitation laser illuminates the sample in this way. In this much area of the sample, it illuminates. Okay, and it generally covers a 250 nanometer emission. Okay, so in the STED microscopy, there is another laser which is known as red depletion laser. So that is a donut shaped laser, laser beam. Okay, and it overlaps with this emission, with this excitation laser. So what it does, it actually depletes the excitation from the surrounding area of this laser. So the effective point spread function of this green laser will be this much. That means only this much area of your sample will be illuminated effectively, and it will suppress the illumination from the uh, surrounding area of this from the center of this laser point. Okay, so this is how the red depletion laser depletes the illumination from the surrounding area from the main laser spot. So this way, the effective point spread function of the illumination is reduced, and the lower the PSF, that is point spread function, higher the resolution. The STED laser suppresses excited state fluorophores located near the excitation focal point by a process called stimulated emission. A donut-shaped ring of fluorophores around the center of the image field is suppressed while the center is not, and the result is a PSF lower than the diffraction limit. So this way you can achieve the point spread function lower than the diffraction limit, that is 50 nanometer. Okay. So coming to the limitations of the STED microscopy. So STED microscopy can present complex optical alignment challenges, but does provide excellent result. So see, it provides the typical XY resolution of 80 nanometer and Z resolution of 50 nanometer. So with this STED microscopy, you can also enhance your resolution in Z axis, whereas with SIM, it was not possible. Whereas the STED microscopy offers better resolution in Z axis as well, and only the most complex thing is the optical alignment, because two lasers have to be aligned in such a way so that this one laser, the excitation laser, remains at the center, exactly center of the depletion ring, okay, or the depletion laser. The speed is medium. It is faster than SIM, okay, but not really fast, okay. And the advantages is it provides reasonable speed. So as I mentioned, that is faster than the SIM, because in SIM you have to capture 3 to 5 images of the same focal plane, whereas here only one image is enough. And the disadvantage is that the system complexity, uh, because alignment of both the laser is quite difficult, and phototoxicity associated with the intense quenching beam. So this red color laser beam, which is the depletion laser, it is an intense quenching beam, and it also creates some kind of phototoxicity to the cell. So if you are imaging a live sample, this may not this may create a lot of phototoxicity, but for imaging the fixed sample, it should not be a phototoxicity is not a problem. Okay, so these are the limitations of the STED microscopy, and here you can see the comparison of confocal microscope image with the STED image. So if these are some microtubules or some fibers, you can see the better resolved structures using STED compared to the confocal microscopy. Similarly, here you can see the better resolved structures using the STED microscopy compared to the confocal microscopy. Here is the another image which shows the difference between the confocal image and the STED image. And you can see the STED image can better resolve the small structures compared to the confocal image. So this is all about STED microscopy, and now moving to the third type of super resolution imaging technique, which is known as stochastic optical reconstruction microscopy, also known as STOM, or sometimes it is also known as PALM, that is photo activated localization microscopy. So how this STOM microscopy works? The STOM activate or excite only a small percentage of fluorophores at one time, reducing the spatial overlap and allowing images with as little as 5 nanometer resolution. So this is the super resolution imaging technique which provides the best resolution up to 5 nanometer. Okay. SIM can offer resolution from 90 to 140 nanometer. STED can offer the resolution in the range of 90 nanometers, and this STORM can provide the resolution up to 5 nanometers. STORM combines the photo switchable dyes and a lower power activation laser, blinking or switching from the dark or off states to emission or the on states. And let me show this diagram, so how this technique works. So let's say this is your sample with the fluorescent molecules. Now generally when you illuminate this sample using the conventional wide field microscopy or the confocal microscopy, what happens? All the fluorophores start giving fluorescence at the same time. And because of that, although they are the point source, they will provide the diffraction limited resolution images, and you will see the entire area gives you the blue fluorescence like this. Okay, so you cannot resolve the molecules at the individual level in the conventional microscopy, wide field microscopy, or the confocal microscopy, because all the fluorescent molecules are giving fluorescence at the same time. So how it will look like if you illuminate these molecules not at the same time but stochastically, means randomly. So what will happen? Let's say in the first image only these four molecules are they give the fluorescence, and computer records the position of these four molecules. In the second image another three molecules gives the fluorescence, and these four molecules are dark now. Okay, so computer will record the positions of these three molecules, and similarly the microscope will take several images in which these fluorescent molecules will blink, means they will give fluorescence for some time and then they will go to the dark state. Okay, so that is known as the blinking of the fluorescence. So the fluorescent molecules will blink and computer will keep recording their positions of the blinking molecules, and eventually the computer will reconstruct the image from whatever the positions it got some fluorescent signal, and it will create the resolved image like this. And here you can, in the super resolved image, you can see there were 1 2 3 4 5 6 7 8 molecules, 8 fluorescent molecules on this line, but that information you cannot get from this kind of a conventional image. Okay, let me give you another example to explain this phenomenon. Okay, so see here it is written as DSTOM, and consider that all these individual molecules are giving fluorescence at the same time. So which kind of image you will see? You will see the image like this. Okay, but you cannot actually resolve what is written. DSTOM is written. Okay, but instead of illuminating all the molecules at the same time, if you if the molecules will start blinking, and if you keep recording the images at different time points, you will see different molecules blinking in different frames. So in the first frame, let's say these four molecules give fluorescence. In the second frame, this another molecules give fluorescence, and you are capturing all those positions of the individual dots, and immediately and eventually you will see the construction of this DSTOM image like this. So this is the super resolved image of DSTOM, and if all the molecules blink, give the fluorescence together, then you can see the image like this. Okay, here I have provided links to one YouTube video, and here you can see nicely how the storm imaging works. So let me open this YouTube link. So in this video you will see how the image of an Eiffel Tower is reconstructed using the storm microscopy. So this is the original image, okay, and in which you will see the blinking of the molecules throughout the structure of the Eiffel Tower. Then here you will see the particle detection at in every time point, and here you will see the reconstructed image. So I am just playing this video so you can see. So these are the blinking lights. Here the particles are recorded in different frames, and if you merge all this frame, you can reconstruct the image of an Eiffel Tower. So this is how the storm microscopy works. Let us see some of the limitations of the storm and palm. So as I mentioned that storm and palm mostly work on the same principle. So in palm microscopy, we use photoactivatable fluorescent proteins. So they are activated at different time points, and their positions are recorded in different frames. So palm and storm offer high spatial resolution, but have low temporal resolution ranging from 30 seconds to 30 minutes, depending on the application, sample, fluorochromes, and the fluorochromes used. So why this storm imaging takes long time? Because see, you have to capture multiple images of the same frame to capture multiple positions of multiple blinking molecules, right? So capturing one field will take around 30 seconds to 30 minutes. Sometimes 10,000 frames are recorded for one particular position of the sample. So that all these positions of the particles in all these 10,000 frames will be recorded, and they will be merged for making the finally reconstructed image. And it provides the typical resolution, XY resolution up to 50 nanometer and Z resolution up to 30 nanometers. The speed is very slow. As I mentioned, multiple images needs to be acquired uh, to reconstruct one storm or palm image. Advantage is maximum lateral resolution, maximum XY resolution. Lateral resolution means the XY resolution. And the disadvantage is spatial fluorophore is required. So suppose if your protein is GFP labeled and you just want to capture the super resolution image, you cannot use GFP for capturing the super resolution image. For the storm or palm kind of a microscopy, you need specialized fluorophores which shows the properties of photo blinking. Okay, so the dye should blink, and you have to use those kind of dyes which gives you the blinking properties. And phototoxicity associated with the multiple imaging and quenching cycles, and imaging close to the cover slip usually combined with the tough mode. So generally this kind of microscopy takes place under the tough microscope, and sometimes it may create phototoxicity in the live samples because of multiple cycles of imaging and quenching. Here you can see one example of the epifluorescence microscopy versus the storm microscope. So you can see in the storm, you can see the super resolved structure of what are the protein it is, maybe some cytoskeleton proteins or some tubulin fibers. You can see the super resolved structures. So coming to the challenges associated with super resolution microscopy. So these days we have several variants of super resolution microscopy which can resolve the structures beyond the diffraction limit. So the diffraction limit is 200 nanometer roughly. Okay, and the super resolution microscopy techniques can provide you the better resolved image up to even 10 nanometer in XY resolution and around 50 nanometer in Z resolution. Okay, but they do have certain limitations. So as super resolution systems image smaller and smaller details, the population of responding fluorochromes decreases, and that requires the development of new fluorochromes with higher quantum yields. So for this kind of super resolution microscopy, we need a better fluorophores or the or the fluorescent dyes for making a better images with high quantum yield. Vibration and spherical aberrations are also a greater hindrance at higher resolution. So when you are capturing the images at very high resolution, okay, at that time a simple building vibration can spoil your image, because see, you are imaging the structure at nanometer resolution, right, in 10 nanometer or 20 nanometer resolution. Even a smallest level of building vibration, if someone is closing the door of your microscopy room or maybe the next room forcefully, then those kind of vibrations can also affect your image acquisition. Some live samples are more adversely affected by super resolution imaging than the others because of high excitation intensity and extended exposure times. So as you see that in all these three techniques like SEAM, STORM and STAID, we are using high laser powers and multiple cycles of imaging and quenching. So that creates high phototoxicity and photobleaching to the live samples. Super resolution systems can eventually reach a point of diminishing returns, where specimen stress and its impact on the data reliability can outweigh the benefits of high resolution. So using the super resolution imaging, you can keep on increasing the resolution, but on the other side, you are compromising on the sample viability and the time resolution. So super resolution imaging systems are much more expensive than the confocal or the wide field fluorescence microscopes. Okay, so they are still very expensive in nature. So these are the current challenges associated with the super resolution microscopy, and I think that's it about super resolution microscopy. And in the next class, I will show you the live demonstration of a wide field and tough imaging system, and we will image the yeast cells in 5 dimensions: X, Y, Z, time, and multicolor. Okay, so see you in the next class, and thank you for attending the lecture.